Welcome back, Biology 104. Today we're going to be covering the sponges and cnidaria notes. So to open up, the question here is, what is full of holes, but it can still hold water? The answer is, you were right, a sponge. So the phylum we'll be looking at first is going to be the Porifera. And there are three classes of the phylum Porifera. Number one is going to be Calcarea. Number two is going to be Hexactinellida. And number three is going to be Demospongiae. These are the three classes under the phylum Porifera. And of course, this will be the sponges. So the characteristics of sponges, they are multicellular, but they lack tissues and organs. Mostly going to be asymmetrical. Some of them will be radial, and asymmetry means they have no symmetry. So it's just, there'll be patterns in a sense, but it's not like one side is a mirror image of the other, like we would think of with like bilateral symmetry. All of them will be aquatic, and most of them will be marine. As they develop, they will be motile, but when they reach the adult form, they will all be sessile, which means they will be attached to a surface. So a larvae will swim to a good position, and then it will attach to the substrate, and then as it grows and develops, it will be sessile. They do have an internal skeleton, and it's going to be made of what's called spicules and spongin, which is just collagen mainly, and that's embedded in a gelatinous matrix called the mesohyl, or the mesenchyme, and we'll look at that as well. The body has pores, canals, and chambers, and those are going to allow for water to come into the sponge, and then it will go out and exit. So basically, they're filtering the water and absorbing their nutrients as they do that so they can feed. Poriferans, for their internal skeleton, you see here in the image, you have spicules and you have spongin. So you can see the difference. It's apparent in the image. Spicules, think of spikes, and then spongin, think of a sponge. A sponge has holes, and then the spicules, they look like a bunch of little spikes. And it's just these little fibers that run back and forth to make this little matrix that you see. So for spicules, their structure is going to be more rigid. They're described as needle-like skeletal elements, and they're going to be mineral-based. So silica is going to be in there. You'll have calcium carbonate in the calcareous sponges, and the function for that is going to be that structural support. And because they're spiked like that, these spicule spikes, they will prevent predators from eating them. So for spongin, it's going to be a little more flexible, like a sponge, right? You can squeeze it, it can bend, and it's going to be collagen, similar to collagen protein that makes it up. So for their composition of spongin, it's going to be more protein, and spicule is going to be mineral. So spongin is going to give it that flexibility, and spicules is going to give it more of that structural shape for the sponge. This is what their body plan looks like. For students who need a visual description, basically what these look like is they look like a cup that has a very curved base, almost like a vase for flowers is what they look like. And on the edges, what would make up the walls of that cup or vase as it comes up towards the top, they have these little canals in them. So it's a network of canals, and then the inner part, just like a cup, would be hollow. It is for the sponge as well. And on the inside, the water pulls in through the walls, through these pores, comes into the middle, and then moves out the top. And that's the general structure for a sponge. And this is what it looks like from a top view. You see the long tubes, and all down the walls of what looks like long tubes or cups coming up out of the substrate, out of the ground, through those pores, it would be bringing in ocean water. For their sexual reproduction, the sperm and the egg are going to meet and form a motile zygote. The zygote grows into a flagellated larva. So although the adult is sessile, they're going to produce sperm and egg that will meet externally um, either on the surface or in the environment. And then that larvae is going to be motile. It'll have a flagella. The larvae has a short drifting stage. They call that plankton or planktonic stage. And then it will settle on a substrate and grow into a new adult sponge. But for sexual reproduction, of course, sperm and egg have to meet, and then the zygote itself is motile. For asexual reproduction, we'll call this fragmentation or fission example. And for that, little buds will break off of the sponge, and then they will just develop into a new sponge. So a fragment comes off, or a bud, and then that will develop into a new sponge. If we're talking about gemmule formation, this has internal buds, and they're produced so that if something happens in the environment, especially a freshwater sponge, and there's a drought, these gemmules can survive for months, and some have lived over a year. So if there's a really long, prolonged drought or freezing temperatures, then the sponge will die quickly, actually, but the gemmules can live over a year. So that prevents, the species from going extinct or the organisms from all dying out. And then when the conditions are right, it'll develop again into the organism. There are 3 canal systems that we see with these sponges. One is the asconoid, another one is syconoid, and the third is leuconoid. And again, imagine that cup shape or the vase. And for that, the asconoid is the simplest. It has a very thin wall or edges to the cup. So imagine a cup with a really thin plastic edges, almost like what you would get from like a fast food restaurant or something, where it's like a really thin plastic cup from like a kid's meal. That would be the asconoid. The syconoid, the wall gets much thicker, and the wall is heavily folded. So what that creates is more surface area, which gives it more canals to move water through and absorb more nutrients. That would be the syconoid. And then for the leuconoid, it is the most complex with extensive branching canal systems that will then have more of those canals to move water through. So it's the most complex. And as you move from asconoid, syconoid to leuconoid, the cavity, which is the spongocoel, that's the opening in the middle, that cavity will get smaller and smaller until the leuconoid, it's the smallest. And the spongocoel of the asconoid will be the largest cavity. So then just to look through some of these structures and what their functions are, like we just mentioned, the spongocoel, that is the large cavity in the center of that cupped structure there, that vase-like structure. Then you have canals. They're going to bring water in. We call that an incurrent canal. And then the spaces between incurrent canals and the spongocoel, we call that radial canal. The ostium, or plural ostia, would be the outside openings into the incurrent canals. And then the osculum is the large opening where water exits the top of the sponge. And then the mesohyl, or the mesenchyme, that's going to be the gelatinous matrix that contains the cells, and the spicules, like we talked about for the skeletal structure there at the beginning. And then pinacocytes, these are going to be epithelial-like cells. So just like the epithelial tissue on human bodies, that covers the outside of us, and this is going to cover the outside of the sponge. Archaeocytes, these are amoeboid cells that have multiple functions, particularly important for the sponge, will be that they help it digest food. We have sclerocytes, or the archaeocytes, and these secrete spicules to form that skeletal structure. And then spongocytes secrete spongin, which helps give it a little bit of flexibility. You have choanocytes. They call these collar cells, and they have flagella on them. And that actually helps create the current that pulls the water into the sponge and then moves it out the osculum. The porocytes are tubular cells, and those, like a tube passageway, pierce the wall and then allow the water to flow into the sponge. So for an image of that, that's what it looks like here. There are those terms there mentioned, and you can see what parts of the sponge that they make up for the sponge. So you see the porocyte there forming a tube, spicules there forming that skeleton, and the spikes prevent predators from eating the sponge. See the osculum, the opening, and then the spongocoel is the large cavity in the middle. There's another image of what that looks like. Now for this one, this would be a simpler structure. This one's a little bit more complex. And then this leuconoid structure last, you see how many canals that there are. So it gets more and more complex. And then here are just some pulled, some of those cells magnified so you can see what they look like. I won't talk about each one. You can kind of just tell what they look like. But the first one up here in the top left, choanocyte, you'll notice it has like this round structure with a little skirt that comes off, and then the flagella that hangs out the back. And that flagella, as it spins, is what's going to pull water into the sponge so it can feed, and it can filter feed on the nutrients in the water. There's another image, and you can see they call that a collar. That's that skirt coming off the body of the choanocyte, and then the flagella coming out there, the flagellum. So then we'll look at the next sponge class. This would be the calcarea, and the calcarea is known as the chalky sponges because their spicules are made of that calcium carbonate, which is kind of like chalk. I mean, that is what chalk has, and that's why you can take it and scrape it on cement, and it kind of rolls off and spreads as a powder onto concrete. That's what you get from these chalky sponges, and these are generally going to be found in shallow marine waters. An example of that would be the Scypha. The next group, or the next class, is the Hexactinellida. And for this class of sponge, they're known as the glass sponges. And as you could guess, they have so many openings that they look, and they are partially translucent. And you can see through them as they have this sort of net, that they look like a net, a cylindrical net, basically. These are deep sea sponges with 6-rayed silica spicules. And the example that's usually mentioned with this is the Venus flower basket. And for that, what makes them really cool is that they are a common wedding gift in Asian cultures. And that's because the sponge itself being hollow and having all these little tiny openings, it can protect small organisms on the inside. And what happens is a male and a female shrimp will actually live out their entire lives together inside the sponge. So it basically becomes a home for a male and female shrimp to have a life together. So it makes a good wedding gift because it's beautiful, but then also it shows this union between a male and a female shrimp. Here's another image of them. You can see in the top left there a shrimp crawling around on the inside. On this next one, see if you can notice there are actually 2 shrimp in this image. You can see this one in the front there. You see it's so translucent. You can barely see that one, but in the background there's actually another one. So that's the male and female in that shrimp couple. The last one is Demospongiae, the last class of sponge we'll look at. And this is called the bath sponge. And that's because they look just like a sponge that you would take to the bath, especially there in the bottom right. It's got all these little cones coming out of it, and it looks like a little bath sponge. These are leuconoid sponges. They have silica spicules and a spongin skeleton. And most of those that have that are going to belong to the Demospongiae, and they call that the bath sponge. So that's it for the sponges, and I'm actually going to stop this and make it a separate video from Cnidaria.